Be happy to try them on different networks and you guys can send you any data logs that just generated.
Pretty good.
At the very least, I can grab the engineering team like directly while I'm here and be like, this is what I'm seeing.
I'll leave you to your thoughts.
Cool. Hey, Veronica, how you doing?
I'm good. How are you?
I am doing well. We're here live at MetroFlow. And
Nice.
we have a studio audience.
Wow.
So there'll be, some amount of goofing around, etc.
Okay. Very
Just,
game show like. I like it.
just so you're prepared. So thank you so much for stepping in a pinch and being the Q&A moderator. Really appreciate it.
No problem. Happy to do it.
And now we've got some attendees. I don't know who shows up for webinars, 2 minutes early. You people are amazing. I can barely get to a meeting on time, let alone early. Nice job, Francisco, Divya, Dana, Mitsu. Nice to have you with us. All right, background still on. Other way. So I'm live from MetroFlow, and today, ooh, how do you get that to appear on camera? Today, I'll be acting as Bob Balderas. And that same storm that knocked out power around where I was at yesterday canceled Bob's flight. So I got to take the BD talk today.
Okay.
All right, we'll get going in about a minute. Oh wow! I just noticed in the Zoom options, there's always been a few like broadcast this live to different places. We can now do live to Twitch.
You can do live to what now?
Live to Twitch. It's like not that I know this firsthand, but if perhaps you're like a professional gamer and you want to broadcast yourself playing a video game, you would put it on Twitch so people can see how awesome you. Oh, I just heard that there are professional gamers in the room with me. Twitch celebrities? Is that
Twitch, so not just gamers, but celebrities.
yes.
Yeah.
Yeah, it's all of my kids' ambitions to someday play games professionally on Twitch for large sums of money as people sponsor them. And then, no, Dad, I don't have to study calculus. I'm gonna just get really good at this video game. That's the way to get rich. This guy did it. So that seems like a fair argument to me, honestly. All right. So I think let me do at least some introductions, and we will get going with. Hi, everybody. Welcome to FlowJo Live Q&A. We do these live webinars twice a month. There's always an intro and some kind of advanced talk. You'll find them on our website. If you found this, you can probably find the rest of them too. Typically for the advanced ones, we try to focus on some kind of very specific high-end topic and do a deep dive, but also sometimes it's nice just to handle all the questions that might come up in a quicker format than sending emails back and forth and whatnot. So John Quinn, Director of Science and Product Development. With me is Dr. Veronica Obregon-Perko, who is going to handle feeding questions in, curating, asking for clarity. Look at that. I am live at MetroFlow and being hosted here. Thank you very much by the Regeneron team with a little bit of quiet space and some of the studio audience here who will be yelling out questions, and I'll repeat the ones that are not too profane and broadcast them to the rest of the group. Looking at the list of attendees in the meeting, I see at least one other MetroFlow attendee here who I can tell is also snuck out and is hiding somewhere in the vicinity around here. You know who you are. Very good. There's my email on the screen behind me, john.flowjo@bd.com. If you have any questions you want to follow up with directly afterwards, that's fine. We had solicited for questions on the website ahead of time, and I do know we've got a pile of something like 12 to 15 questions that came in that Veronica is going to feed into the Q&A if things get quiet. But I definitely want to keep it moving by answering the questions live for the people that are here and present, asking them. We've got both a chat for the webinar, and you'll see there's a Q&A box down below at the bottom of your screen. You can type questions into either. Veronica, do you have a preference of which one you're going to? Actually, I will have an eye on the chat. Maybe if Veronica keeps an eye dedicated to the Q&A more so, and we'll go from there, trying to handle all the live questions. And then if we get quiet, we'll pull ones that have been sent in ahead of time.
That works for me.
Cool. Using FlowJo 1010 to answer the questions that are FlowJo related, which we're going to hope like 90% of the questions in FlowJo Live Q&A are related to. Maybe we'll get some BDRC questions, which is okay too. I usually have an includes list that's going to, almost aimed that right. That lists what topics we're going to hit, but for today, it's open Q&A, whatever everyone wants to talk about. So with that, I'll share my screen and we'll start doing questions. Sorry, always lots of choices about exactly which screen to share. We'll just go with all of it. And let me pull the chat up so it is still visible. And close down Zoom. Okay, so I also just have, as I expect, a decent number of questions will pertain to, I've already clustered, or I've used some kind of dimensionality reduction. So I just pulled one up that I did something with, so that we have it ready if we need to. All right. Perhaps, Veronica, why don't we start off with one sent in from the webpage.
Absolutely.
Break the ice.
All right. So first icebreaker question is, can you please explain how to use the parameter scaling preferences for cytometers?
Okay, can do. So in FlowJo, the heart in the upper right-hand corner of every window is the preferences. If you click that button, there is a cytometers tab here. The cytometers tab takes you to this interface where every cytometer you've ever added data from is listed here. If you've only added data from one cytometer, you'll just see one listed. We have preferences built in for every cytometer, but we'll only display the ones that you're actually using just to make it easy to find the ones that are most pertinent to you. I have attempted to use every cytometer, so my list is fairly long here. Whatever's highlighted is just what you're looking at the preference for. It is not necessarily the preference for the cytometer of data that is currently in the workspace. The things that you have control for here, and what, I'm trying to remind myself, what cytometer did this data come from? I think this is an A5SC. That may matter on the advanced screen. So let me pick the A5SC out of this list. Okay, things that are perhaps interesting to you, you can adjust the log and lin scale. At Claim, we've done a really nice job working with all of the different vendors to make sure the default preferences for scaling are good, that they've given us images of what their data looks like on their acquisition software. We've tried to match that as close as possible in FlowJo. So if you're using a cytometer with all the default settings, it should look good. If you've adjusted them all, you may want to change some of this. There's also an enable transform button down here in which you can turn on a transform. We'll try to figure out which transform makes the most sense for your cytometer. Typically, we use a bi-exponential transform unless it's something like a mass cytometer where arc-cinch is better, Miltenyi where the Miltenyi transform could be used, and so on and so forth. There also are parameter filtering options down here. So none are being used on this particular cytometer, but if I click on maybe like one of the Coulter ones or one of the A5SC that I've set up some parameter filtering on, no, not A5SC, sorry, A8 is where I've set up the filtering on. You'll see that I've chosen to hide some parameters from display. You go about by just hitting add, type in some string of text that you'd like us to search the list of parameters for and hide anything that has the string of text. Some ors and nots can be used to make a more complex hide this, show this, here's an exception list, so that if you've got data that has an enormous parameter list, maybe you've got -h's, -w's. With the A8, you might also have -t's that don't really add anything to the analysis. You can just choose not to display them so that your lists are more succinct when you're trying to find a parameter somewhere. The other bit that I mentioned I was going to show was just advanced rules. If you hit advanced rules, and actually I meant to specifically be on the cytometer that I was using, so it would be able to preview some data. Rules are from a different cytometer. I guess that wasn't an A5SE. Anyway, good enough. You can set up advanced rules over here, in that if you hit add a rule, call it something, new rule's good enough for now. You can add a condition where the condition looks for some kind of string. You could say is fluorescent, is compensated, etc. It'll highlight over on the right any parameters that match that condition. And then if you hit the transform button, you can adjust the data as needed based on the appearance right here, and will remember that preference for everything that was listed in that condition. So that gives you the ability to do things like come up with preferences that are as granular as forward scatter and side scatter, have different default scales. It allows you to do things like set a suite of imaging scales for A8 data or S8 data to do with whatever you want. One other small advantage in this window is that it actually allows access to all the different controls here. The plus to expand the range and the positive decades you can see move together. We've always assumed in the past that if you had more decades, you would have more range. If you had more range, you would have more decades. Not true for some of the newer cytometers that will show a 7-decade range, but actually only really display the data over 5 decades. So you could do something like add display out to 10 to the 7th, but still resolve down to showing 4.5 positive decades to keep the data looking like not so squished or whatever. So that's useful. That's useful particularly for S8, Aurora, some of the Novasite cytometers and so forth. I'm just going to cancel this as I don't think I needed these preferences. A5SE data scales by default really nicely, but just so you know it's there. These advanced preferences can let you get as granular as you like to go in setting up how all of your data will be displayed by default. All right. And the chat is still empty at the moment. We've got a bunch of passive listeners today, but the list is growing. Awesome. We'll start knocking out questions that were emailed in ahead of time. And hopefully I will say, I don't know, something either controversial or confusing enough that that'll generate some chatter in the web chat, and we can answer some of your questions live. All right, if there's no further questions on scaling, Veronica, why don't you hit us up with whatever the second questions that was emailed in ahead of time.
Sure. There is actually a question on the same thread of scaling and transformation. What transformation is best for my data, and why do I sometimes have negative values in my data?
All right, so more of a hand-waving kind of question. The transform that's best for your data will depend on the dynamic range of the data. So if you've got something like forward scatter, side scatter, DNA content, there's not a huge difference in the smallest and biggest values, a relatively small dynamic range. We can use a linear scale for that kind of data. If you put that on a linear scale, you can, linear is just the best for a number of reasons, including every tick mark on your scale's got the same value. It's easier to interpret. It's easier to just intuit statistics. That looks twice as big. Works. That kind of interpretation is good. You can go below 0 to negative numbers if you want. So that's nice. If you get into anything fluorescent, mostly antibody dependent, is that actually that's the qualifier. You're going to have a very large dynamic range. Things that do bind your antibody might bind at 10, 100, 1,000 times as brightly as any kind of background signal you get, which makes it difficult to interpret on a linear scale. So typically folks have used either log, but log cuts off, like there's no 0 or negative numbers on logs, that's got a limit, or some variation of the bi-exponential or arc-cinch transforms that will compress the data more and more the farther you get up the axis. And since things that are really super duper bright, we care less about finer small differences in, if you've found 1,000 times the volume of some negative or background population of CD3 versus 1,010 times, still positive for CD3, very bright for that. So visualizing that as one coherent population compressed a little bit more It's fine. It's nice. So for example, we could go to one of these full stain files. Maybe I'll go to my concatenated one since I've got that one loaded in and pull up any kind of fluorescent parameter. All right. So all this that I'm talking about is just if it's bi-exponential, you'll see a 0 because the bi-exponential scale compresses the data more at the high end by applying some kind of log-like transform at the high end and just leaves the bottom as linear. The benefit of the linear transform at the low end is you can see negative numbers. So if you've got a population that revolves around 0, it doesn't get cut off, which is also the answer to the second part of that question. Why do I sometimes see negative numbers? If you've got a cytometer that has done baseline subtract or something like that, or a population that's fluorescence is around 0 and then you compensate, or near 0 and then you compensate it and there's a little bit of subtraction applied, you will see a population that distributes on both sides of 0 as this one does. If the skew is a little bit more to the left, you'll end up with a negative MFI or a negative fluorescence of some portion of that population, even though it's totally properly compensated, acquired, etc. Fluorescence data is relative, depending on how much voltage you put through your PMTs or whatever you've got, collecting photons on your cytometer. You can move any signal up and down, and we tend to end up compensating and zeroing negative populations back down near 0. Sometimes they go below. That's all that comes down to. Okay, and if you wanted to change a transform on the fly, it's the T button, customize, and that allows you to either add some space, so this looks just like the advanced scaling tool, or to increase or decrease the width basis, which effectively puts more of your data into the linear range, allowing you to visualize a wider range and also minimizing the variance across your negative populations, which can be a nice thing if you're doing any kind of clustering or dimensionality reduction tool, and that frequently minimizing variance of the negatives helps you take that out of being the focal point of what the clustering algorithm identifies and focuses on as interesting information. Okay, and apply to make it happen. All right. Other questions?
All right, we did have one come through in the Q&A box. It may need some clarity, but let's see. It says, hello, if I acquired my samples in the spectral mode, can I analyze them as conventional, or do I need to re-export them from Diva?
You can analyze them as conventionally. I think probably the most important part of that question is the idea that spectral data versus conventional data will have different analysis techniques. They will have different compensation versus unmixing applied to them. The real difference in that data is that if it's conventional, it will have had one detector for every fluor you've measured. If it's spectral, there'll be many detectors and fluors intermittently spaced out. And so when we unmix spectral data, we have to attribute all the signal from a given fluor into a parameter, even if it comes from many different detectors. Similar actually what traditional compensation does, except you're just taking relatively small amounts of signal and reallocating them back to the primary fluor. At the point it is unmixed, you just have targets with an intensity, and you can use all of the same tools that you might use to analyze any other flow data on that spectral data. Some part of that question might have been specifically with the export format that came out of a spectral instrument. I know the BD instruments allow you to export as an experiment, export as an FCS file, etc. If you've unmixed on the instrument, we'll put the compensation matrix into those files in different ways. Generally, you can either just use the matrix that was exported from Diva. You can make a new one in FlowJo if you've got the single stain controls. You can start from scratch. Editing that matrix could be a little trickier depending on how you've exported it. We generally recommend exporting as FCS. So I hope that got the question. There was a couple of possibilities there. I think I answered them all. Let me know in the Q&A if you had more questions.
Absolutely. And if that user wants to clarify in the chat or resubmit another question, please feel free to do so. But while we're on the thread of spectral data, how do spectral plots work in FlowJo?
Okay. Let me. Okay, good. That one actually has some data. Let me pull up a spectral plot. So we've got two options. We've either got the spectral plot, which is intended to show for a given sample or a given population on a sample. You can go down to good events, perhaps. Oh, I didn't bring in the QC on these. All right, so we'll stick with the top level. or we can make a population. If we want to demo that, we can even look at the differences between. Sure, population. So looking at a specific population, hit spectral plots. Sorry, pick a specific population, then look at spectral plots. It'll ask which detectors you want to look at. Typically, it's all of them. And you will get a visualization of your signal versus each one of the detectors. Okay, if you have gone in and modified the transform so that you've got different transforms on them to visualize them, it'll ask you to unify. So let's do that. Let's go up here, something like that. Okay, unified. Cool. So visualization enabled. So the gist of this is now that you can see the signal coming out of some given cells across all of the detectors. I think the default view, which is showing everything, is a little much. It's harder to resolve how bright some of the signals are compared to some of the thinner signals. And so I tend to go right to the visualization tools under options. You can configure this. Sorry, my zoom controls are right in front of. Let me see, can I move these? Yes, I can. Cool. I can move these to just go to, how's this density plot being shown? I think typically hiding something like the lowest 10% of density is nice to do. Focus on where we actually see signal. So what we're showing by default under the options is the density and bands. You can turn off one or the other or both and go to violins, etc. So it's just giving you an idea that you've got a lot of signal out here in YG602. You've got another peak down here around the B710, 750 area. And that is where the concentration of the signal is. You could gate on this if you'd want it. So if I wanted to say identify, perhaps a positive population within one of my controls, I could do so via the spectral plot. At this point, creating a new population, I'll call it population. So not super creatively named, but there we go. There's our population gates that are now being acquired right here in the workspace that could be selected when we get to compensation if we want in the comp controls. Your other viewing options, so I'll put the bands back on and turn off the density. And all of these options can configure the width of the bands and so forth, how these are formatted. But if you prefer the line plot view to essentially see where the brightest spot on each one of the detectors are, you can do so. There are also violin plots so that we can do that, and you can maybe turn off the bands to go with that so we can see the range and distribution of the cells expressing the signal. I think the important thing to keep in mind with these plots is that you can imagine almost they're like a histogram turned on the side and just shown as a heat map across a specific range of detectors. Doesn't have to be fine, it's okay. I've announced for live, there's a studio audience, you can jump up and down and yell, and also the signaling in the Macs is really good at filtering out noise. Do what you will. Okay. That's all right, it won't come up, it won't come up. No, we've got a virtual background on. So hi everybody, that's the joys of doing a live presentation. Interaction, nice to have some. Okay, so those are the configurations for the various views you can look at. There's places to copy content, etc. You can turn off the grid line, the zero line. You can name the populations, something more specific. So that is the population viewer. So the spectral plot, the population viewer is one that, and we actually select something again, that actually allows you to do comparison between different populations. Click that, use all the detectors again. Okay. So there'll be a lot of similarities, particularly in the graphics and how we can configure the appearance. So same bit with options of showing whether we want bands, violins, density, etc. I'm also going to configure this and turn down the density, probably like again to something more like that. Okay. Interestingly then, you can also go in and if you've got some other population, just drag and drop it here and we'll see the overlay. And we can compare where do these two populations co-express, where is their separation, where is their differentiation. If you would like to use a specific autofluorescence population, don't have one handy, we could probably bring one in. Doesn't seem like we're piling up with questions. We could take a little more time with this if we wanted, but imagining we had a specific autofluorescence population, we'll start there and just say, just drag it and drop it and use it as the negative. That would allow you then to do normalization versus that specific autofluorescence population. So at this moment, I'm just comparing total fluorescence versus lymphocyte fluorescence, where there was a lot of lymphocyte fluorescence. Since I am, let's also just change this to showing only the population names. So when I say lymphocytes, it comes in as lymphocytes. Anyway, just showing me where there's a lot of fluorescence, in this case, for all the cells other than the lymphocytes. So anytime you want to do some sort of differential expression of background fluorescence where you have multiple populations, this is a tool that can be used for that. Other tools that are available in specifically the comparator tool, the spectral population viewer, along with normalization, is a similarity matrix. And so if you've got a couple different populations in here, you can calculate a cosine similarity value for those populations, and that they're going to be resolvable across multiple detectors, tabular form or chart form. So with 2, that's fine, relatively small. You can do more if you want to. Okay, I think that is the gist of the population viewer and the spectral plots. Let's see. I do see we've got a question in the chat. On HT data on the plate display, is it possible to have the percent of whatever population you are displaying? If so, how? On the plate display, okay, so I think the plate display would refer to, we've got 2 ways we'll show plates in FlowJo. Let's open up a new workspace and drag some plate data in. So data, plate data, cool. Unless I missed. Okay, we've got a couple of different plate views, and let's also do edit columns. Let's show the well ID and organize by that. Okay, there is the plate editor tool. If you do plate editor, that will allow you to read the samples and attributes from the group. Everywhere where you get a circle, that is where we found a data file that matched the well ID for this display. The 9 in the bottom of all these is just indicating we've got 9 keywords that have some differential information. So this tool is mostly used for annotation purposes as opposed to statistics. So most I'm just pointing out that this is here as a way that you could either add some new keyword by giving it a name, new with a B, for being silent, and a value. And then you can just drag and drop this to anything that you'd like, either a row, an individual well, the entire interface, etc., column. Or you can do edit file and import an entire spreadsheet of keywords to line up. Some of this stuff can then be heat mapped in here. So if you had a keyword that had mentioned the number of events, you could do that. News just showing 1 for everything in 1 column, so we'll just turn that yellow. So that's possible via keywords and visualization on the plate tool. We also have some plate information available in the plate-specific graphics to show information in the layout editor. So if I would say, make a gate, sure, that's fine. Add that to the group. We could display things like percent of cells, counts, whatever, in a plate-based format. So I'll do edit, workspace, statistics. Let's do frequency of total, and I'll add a stat for count. Okay, within the layout editor, the L at the top of the screen, this is the plate view. Drag plate statistics here. So you can take whatever stats you want, drag them in, and we will get a heat mapped version of those statistics right here. If you click on the plate tool, properties panel on the right includes what you want to show as the heat map or the upper half of the heat map. Just make it a specific heat map. You'll get numeric values on the side. So in this case, you would be able to estimate, like, okay, the yellow means something like above 8,000, etc. If you actually want a grid with specific numbers, I would probably actually do that in the table editor, which is less graphic, more numerical, and maybe just batch and iterate by column and row to make it plate shaped if you wanted to. If you don't care about that, we'll just do that now for frequency and count. Actually, let's just do one at a time. If we want it to be 96 values, frequency of total, create a table. There is the percent of events in each well. add in keywords such as well ID if you want it to be more specific, but I guess the name of the file also includes the well ID. Okay, so that was like 3 different ways to show information about plates. Hopefully, at least one of them was close enough, Lola, to what you were hoping to achieve. And I'll mention again, if you would iterate this table by rows or columns of your plate, you could actually get it shaped like a 96 well plate, and could also send this to the layout editor if you wanted to put this next to the heat mappy view or any other kind of graphic. So I could do, instead of going to display, send it to the current layout and send it over there. We'll have both of those things, some kind of numeric values alongside a graphical annotation doing heat mapping. Okay, this is more or less what we do. Score. Was wondering if there was a way to display the percent on the heat map. You know, the other thing I didn't mention was if you really wanted heat map and a percent, is that you could go in here and choose the heat map option within the table so that when you batch this thing, then it is both heat mapped and has a number. And again, I would probably just, I don't know if this data has, I could add keywords for row and column if it doesn't already had it, but if so, then I would just iterate by and choose the keyword like row and column so that it spaced out 96 if you wanted to, you know, have the correct shape so that it's easy, an easy reference to like look at the plate, look at the stats. All right, got a cool win. All right, shall we move on to the next question? I think I saw the Q&A was starting to light up.
Absolutely, yeah, very cool. I feel like I learned some stuff in that segment as well. Okay, so we have actually 2 that came through in the Q&A. I'll start with the one that came through first. References the Beckman-Coulter cytometer, but I guess could be applied really to any. But the question is, in FlowJo, the fluorescence intensity of markers appears lower than what I see in the cytometer software, and I see variability in the positive percentages. Have you ever encountered a similar problem?
Gotcha. Likely that comes down to an issue of binning and perhaps scaling, because we do adjust binning with scaling. Usually the differences are pretty minor, but if you collect data on a cytometer, the typical range is it's divided up into 262, 144 discrete channels. And so if you've got, and then we convert that to whatever your intensity range is, it's essentially an interpolation to say if you're in the highest bin, that was your maximum intensity, it's 10 to the 5, 6, or 7, or whatever your cytometer can do. If you're in the lowest bin, then you're at 0, or if you're going negative, some small number. If you trim out some white space, you know, sorry, first, if you bring it into FlowJo, graphically, we will break it down into 4096 bins. And so, you know, it's possible that a few cells, statistically, we do more than that, not quite 262.144, but a smaller version of that. So if you're calculating on absolute raw data on the cytometer, there will be a little bit of a binning effect that lumps together similar events, more so in FlowJo than it does on the cytometer. And then also, if you would trim out white space at either edge of your data, we'll still use 4096 bins to show you your graphics, to decide what's in or out of a gate, etc. But now there's a smaller range. You've cut off a little bit of essentially unused space, so you'll see small differences in numbers there. We typically feel like those numbers end up being less than the measurement error associated with doing cytometry, so we're okay with that. It does mean that if you are working in a clinical lab and you need to see exact numbers all the time, perpetuated all the way through the experiment, it'll be important to have your FlowJo workspace as standardized as possible, and adjust the transformer at the beginning and never again type things. Fair, and feel free again for clarification questions. There's space in the Q&A if I didn't answer everything you were wondering about.
All right. Okay, so going on to the next question related to compensation. How bad is it if I put the unstained sample as my universal negative if the majority of my single color controls are made with beads?
Bad. So that is it's a good question, in that it's important just to remember the fundamentals, that what we're attempting to do when we do traditional compensation, and let me close up this one with plates. Let's just at least bring in a handful of comp controls, new workspace, and we can talk about this with pictures, because pictures make everything better. Okay, and sorry, I'm being a little flip, but it is a good question, and it's something people ask all the time. I'm just bringing the PFIX data just mostly so that I can show off Tim's crazy photobomber face that I've appended to his own folder, his fault. Okay, I'm going to go to compensation, open this up so I can talk about plus minus gates with a picture of plus minus gates. Okay, so here's just a picture where we're doing traditional compensation. The idea behind traditional compensation is that for a given color, I want to show the algorithm cells that have actually bound to this particular color and then background. So the reason for this is that so I can look at the median fluorescent intensity of all the cells in this green population here and know that they've only been stained with APC, or APCH7, sorry. Everything else, it just hasn't been added to this tube. So we know that we would like the contribution to be effectively 0. However, there's always noise in everything in our systems, a little bit of electronic noise, a little bit of autofluorescence, etc. So we don't want to compensate down to actually 0. We want to figure out for this type of particle, what does background look like? And then we'll use this negative population to estimate background and then try to make the median fluorescent intensities match. If we have a different kind of particle for background and single positive, then we're aligning to something that could be brighter, could be dimmer, and we will probably either over or under compensate, and it'll be tough to know ahead of time. We'll see data spill into the axes and so forth. So the alternate approach to compensation is autospill. If I flip on autospill, that's going to do, all the gates just disappeared. Yikes. That's going to do compensate. Well, other than the cleanup, that's going to do compensation without those plus minus gates. Instead of making the MFIs line up, we'll do linear regression across the full range of the data. And then just try to make sure if we draw a straight line between the parameter that we've got and anything else put on the y-axis, and we'll do each one, that line should be flat. So if you have reached a situation, we've got a set of comp controls, and you perhaps, don't have, a good negative, you're going to use beads as the negative. If you do have a range of fluorescent intensity, it is possible you might be able to use autospill to get an estimate of the distribution of the data and proceed from there. That to get back to the primary answer is you've got to match the type of particle for a given control. Between different colors, like for AX48 or AX647, these two could be different particles. And in fact, in this experiment, you will see that we've got a live dead marker that's being run on cells and some beads for some of the fluorescent markers. That's fine, because for a given color, you're still using the same particles to figure out what is background for this target positive population or an autospill, or the full extent of the data. Okay. So follow up with more questions if you have any. It looks like there might be one more in QA.
There is, also along the lines of spectral compensation. Let's see, it says, when I acquire in spectral mode, I should put my autofluorescence channel as my true negative. Does it affect compensation the way a universal negative would? If not, can you please show how to set the autofluorescence channel in the correct way in the compensation window? I feel like I'm doing it wrong because the values on my matrix are always very high when I'm looking at my n by n plot.
Okay. Yeah, so I think that question comes down to, if I want to do autofluorescence extraction, like what's the negative? And I will mention, let's go find some data. Here was the one that I wanted to use. It's going to pull it out of here, I think, because there is somewhere in here some comp controls with some universal negatives. Okay. And some autofluorescence controls. Okay, yeah, there is. So in this data set, we've got an autofluorescence file, we've got an unstained file. I'll imagine they look very similar, but there is an option for using true 0 as the negative if you're doing traditional compensation. So if we hit compensation, just going to pick all the channels. So that reason for that box popping up there as an intermittent step is FlowJo won't know ahead of time if you're going to do spectral versus traditional. So just in case it's traditional, it's saying, hey, would you like to carefully pare down the channels you actually used? If you're doing spectral, just say, nah, select them all. Easy peasy. Okay, scrolling to my, let's just also remove the unused channels. Okay, scrolling down to my autofluorescence. At the moment, it has the, FlowJo, even though the file's called autofluorescence, it doesn't know especially what that is. So it's just given it the universal negative is the background. We can actually go pick out of this list something called true zero. And what that'll do is use what's effectively going to be, a unlabeled, pretty much backgroundy type population. If we go find it, UV450. There we go. So you've got something like barely any signal in it. And so if you're doing traditional compensation, just like I had mentioned on the previous slide, we have to have some kind of negative to say, okay, whatever signal we see here, we're going to attribute what we've brought into what's been applied to the cell. And this cell, it's only autofluorescence, and a tiny bit of electronic noise. And so what's our goal? What do we set it down to? And so we can actually just use actual zero as the negative in that case. The alternate approach, of course, is to use auto-spill. You don't need gates. Linear regression just attributes any deviation from a flat line to being the contribution of autofluorescence. You just go from there. And so you don't need to have a negative population. Either of those approaches will work, but they'll work a little different in that if you're using true zero, you actually are trying to bring something all the way down to a zero background for the autofluorescence contribution. You tend to get a little more signal attributed to autofluorescence than if you use the auto-spill method. I can't say if one or the other approach is better. I can say that most of the cytometers, we might be the only people with auto-spill at the moment. So most of the cytometers are going to use the true zero approach. So if you want the numbers to match from FlowJo to what you would see if you ran it on any other acquisition software, the true zero approach would be the way to go. All right. And let me know if there was a lot in that question. Let me know if I correctly got it and answered all the parts of it. But those are largely the mechanics right there. Oh, and check the spectrum if you're doing spectral. Perhaps the single most important step.
All right, so I'm not seeing anything else or any follow-up coming through on that question. So I'll move on to another, but we can always circle back if there's follow-up. Next question. What's this BD logo button inside FlowJo?
The BD logo button here is the link to the BD research. I like that. What is this BD button? It's the BD button. It just makes BD magic happen. Sorry, it's the BD research cloud. Online tool that you can use for a variety of functions. It works with your portal login. It's free up to a certain amount of data storage. So all the tools can be used if you've got a FlowJo portal login just right off the bat, just by typing in the same password that got you into FlowJo. The primary features of the BD Research Cloud are that you can do panel design in here. You can create Diva experiments based out of, preferably, the panel you just designed. You can put together a workflow where you can assign different tasks to people. You can have data that's stored here that then you can access directly from all over FlowJo. So most of the file open and whatnot options include open from the BD Research Cloud, which just if you signed in, takes you back to a loader screen that's pulling out whatever data is here in the Research Cloud. You can upload cytometers that when you do panel design, sorry, there was the data load screen popping up over there. So when you do panel design, it can be either from pick an off-the-shelf configuration of a cytometer, or add a config file. So that is your specific cytometer. The off-the-shelf configs, I think, are limited to BD machines, but the uploaded config, so it's my specific cytometer, is anything that puts out a config file. So you can do it for any cytometer. It's spectrally enabled. It includes some suggestions if you're on the S8 for doing image, if you're doing an image/fluorescence acquisition of how to choose image fluorescent parameters to use. A few other things built in here. It's got an inventory of reagents. You can track that if you want. It's got populations that you can define. So if I think T cells are defined by 348 FOXP3, et cetera, when you're going to build a panel, you can say, please bring in my Treg population markers to start with that as a backbone. And it's going to be a big panel, so let's mix it in with some B cell markers, et cetera, and put together a panel in that way. You have the ability to organize users into groups and give specific permissions to who should see what, organize things around projects. And then there is Veronica's image actually as our default user. And so you can go in and set up your own user profile there. Okay, I don't want to turn this into a full BD Research Cloud talk, but it is fully integrated into FlowJo. So if I add, data from one of these panels here, it just pops up into my FlowJo workspace. I can save back up to the BDRC. It's got some capabilities where if you are doing imaging data, it can smartly extract only the images you're actually working on down to your computer to do further pre-processing and so forth on that. Highly recommend. We just did a webinar yesterday. I believe it was yesterday on the BD Research Cloud. I'll just point to FlowJo home. Again, you found our webinars already if you're listening to this one. But under webinars, I guess it would have already moved to the recorded webinar since it was yesterday. Intro to the BD Research Cloud happened there. If you want more on that, check that webinar out. Emily, who is the product owner, does a really nice job going through what all the functionality is. Okay, we have got about 17 minutes left of this one. We look like we're doing pretty, oh, there's one in the Q&A, I think, and the chat is clear. You want to hit me up with the Q&A one, Veronica?
Absolutely. Here we go. So our ND asks, would you recommend using the downsampling option if some of the samples did not get to the number of events required for the stopping gate? If yes, can you show how to use the downsampling option?
Yeah, maybe. So it's flow cytometry, and we will tend to collect tens of thousands, if not hundreds of thousands of events, even per population, maybe millions of events per file. If you end up with data files that you're going to try some, comparative statistics, automatic processing like a clustering algorithm or whatever on, it's nice to have reasonably similar numbers of events. If you've got one file with 1 million events and another file with 10,000 events and you create a Tisney plot, well, then the file with 1 million events will completely dominate what populations appear. If you end up with a bunch of different samples and you've got 30,000, 32,000, 33,000, 34,000 kind of events, close enough, doesn't matter. So I wouldn't bother for such small numbers like that, and that, your data is precious. It sucks to throw any kind of data away. The more you can use, the better. We're using cytometry in large part because we want to be able to collect huge volumes of data and be able to find rare populations and so forth. But if you want to downsample, there's a couple ways to do it. One of them, for a specific population, we do have a downsample plugin that's pretty simple. Just have to, under Workspace and Plugins, choose it from the list. If you don't have it, you can go back to the Exchange. The Exchange is just where we house plugins. Plugins are generally tools that, we found somebody doing nice work, collaborated with them, and posted a way to use maybe their R script or whatever it is in FlowJo. They're all free. If I was going to run downsample, I'd go here to the downsample, hit the download, a file downloads onto my computer. Hey, and you can get a periodic table of plugins. Thank you, advertising. Anyway, it comes down to my computer. I stick it wherever I want to put it on my computer. I have personally chosen to put all my plugins in applications, and I have a folder that I creatively named plugins. And so here's all my plugins. They're just .jar files. So when I start FlowJo, then anything that's in that folder appears in here, including downsample. A handful of the plugins require R because that's the language they're written in, and we use some R functionality to make it run. If you get one of those, it'll come with instructions on how to install it. Downsample doesn't. Yeah, just have to download it, start it, hit the button, tell it how many events you want. I'd like 10,000. That's that. You can drag that tool to the group to have it fire off on a whole bunch of different samples or populations, the exact same downsample, that'll work. Alternately, if you are in the process of setting up some kind of higher dimensional workflow, you want to either concatenate or maybe export or something like that, a specific number of events, you can go pick multiple populations. I'll hit select equivalent nodes, right click on that, export/concatenate. So a lot of the operations that would precede downsampling are some sort of export or concatenate to handle a bunch of things all at once. And so if you're trying to even things out, you would say include no more than, and pick a number out of here. It so happens that I've collected exactly 500,000 events for all of these files. If there was some variance, the number that would appear by default would be the smallest of the numbers out of the populations I selected to ensure that I could get to that number for all of them. You can type whatever number you want. And then when you put them all together or export them, so concat or export, it'll just do the downsampling as part of the process. And you don't need to do a separate step. Either one of those things work exactly the same way, and that is a random downsampling. Just something that we're looking into is different ways to downsample with either some specifications to it or some density dependence, a tool for the future. But for now, 2 options to get a downsample population, and whether I need to do this or not, I think depends on how different the number of events being contributed from each file are and how important you think that may be. Also, something to consider. Imagine you had 100 files and 50 of them were from healthy people and 50 of them were people that from a disease you were trying to see what the biomarkers were from it. What may actually matter the most is whether the composite of events you took from all healthy people and all diseased people added up to roughly the same number, maybe more so than each one of the individual samples having the exact same number. If that was your true dividing factor, then some randomness of people, person to person variation probably matters less than do I have a representation of what healthy versus disease looks like holistically. Okay.
Great. Okay. So I'll go back to the pre-submitted questions while we wait for something else to come through on the live event. Can I use Cluster Explorer only on populations that are algorithmically defined clusters?
Okay. No, Cluster Explorer as of version 10.10 has been expanded so that you can actually use it on anything. This concatenated file I put in here had a variety of different just categoristic features implanted in the data as like a 1 or a 0 or something that I gated on. For example, let's go and pick one like age. So binary distribution of age, old people versus young people. No comment on where that dividing factor is. Just put some gates on it, called it old and young. So there's just gates. These are just range gates built in. They could be anything, rectangular gates, polygon gates, etc. You can represent any of those things within the Cluster Explorer to do comparison of how do these tie into my clusters. So if I hit the Cluster Explorer button out of algorithms, it's asking me what clustering result do I want to use. I had run T-Rex on this data. T-Rex also creates dimensionality reduction parameters as part of its process. So I've got some T-Rex T-sneeze. So I'm going to deselect the other things, the percent change. We won't look at that here. And then it's asking me what parameters I'm interested in. I won't worry about this too much, but right off the bat, I'll turn off live dead and autofluorescence as things that I want to carefully examine. Up here, I can show populations as a new cluster set. I can also turn off the ignore nodes belonging to unselected clusters. And when I configure this, let's add in those ones that I gated on. So we also have a binary for male and female as a gate that are in there to see differences there. And say accept. Cluster Explorer does its thing. It's going to pop up a whole bunch of windows that are just different ways to view clustering results. Okay, there we go. Just moving the main workspace aside. T-Rex, incidentally, across a whole dimensionality reduction plot, identifies the little pieces of it where there's real differences between some kind of category. So hence the sort of the funny looking clustering result. You can almost imagine that there's a much larger t-SNE plot here, and we're just highlighting the parts of it that are different between young and old, etc. Okay, so there's all that, and we're looking at all of the different clusters that algorithm produced. But what if I want to focus on some of the gates that I had drawn on there? Here's the cluster set. So by default, it's picking T-Rex because it was the biggest one, but it could also just pick that populations one. And now I've got female, male, old, young, and we are able to differentiate the data based on these categories. It doesn't look like there's tremendous difference, although maybe right here and right here, because we can see if we use the Cluster Explorer to mouse through any of the different categories, that most of the t-SNE plot shows up. A few spots where there is a real difference, though, that we can see. Like specifically, it looks like whatever that population is there, and we would probably actually want to look at a cluster as opposed to just all of the cells that came from women. But we go find out what that is. Maybe we'll be going back to our T-Rex clusters, see if anything was right there. Hey, there is. Well, no kidding that one came up as one that had a difference to it. And we can identify this as cells that specifically look like relatively high on the Keller G1, a lot of CD5RA, 45RA, or CD3. Well, that just makes sense if they're also expressing some of these other things to try to figure out what they are. There's the heat map version of that as well. So at any point, you can go through and use a gated population as the things that appear in lieu of clusters. You could also go through the T-Rex clusters and filter down one at a time by female, male, old, young, whatever demographic it is that you've embedded in the data through some kind of categorical information. Okay? Closing that up.
All right, we have a question in the Q&A related to high dimensional analysis. Can you show us how to analyze flow data using t-SNE?
Sure. Let's pick a relatively small population. Let's just pick old people. I'll go t-SNE. Click the t-SNE button. It is built into FlowJo for a while now. So all you have to do is find the in-app button. The top portion of the box is just what parameters are you interested in? In many cases, it'll be all of them, but probably not every single one. We can imagine in this data, if we have gated down to CD8 T cells, that there's no longer a lot of difference in the data on things like CD3 content, autofluorescence, CD8 content, probably CD4 content, because they're all negative for that, live dead. So anything that you've already gated on, you may want to remove. The idea here is that these algorithms are going to try to put together things that are alike and separate things that are different. If you add in all of the parameters you've already gated on, so all the cells have pretty much the same signal flow, when you're telling the algorithms that these cells are all alike, push them together, make a worse result. Probably don't want to do that. For something like CD3, which maybe does have either differences in intensity related to things like stimulation or a challenge. Maybe you want to leave that in. That one I'll leave as your call, but I'll take a few of these out. And they are T cells, so it's probably true that some of these other markers aren't going to be very exciting downstream here. Yeah, I don't know, I'll take just a handful of those out. Anyway, and say, okay, I've got some choices down here, auto or manual, and then down here, exact or approximate. I'm going to pick approximate, and then Barnes-Hut versus FFT. I'm going to pick FFT. I'm going to hit run. That's probably going to take, I don't know, 2-3 minutes to run or something like that. So I'll just get it going while I explain those choices. OpsNE or auto versus manual is just, how do I want to set those defaults? If I'm going to do it as, well, number one, choose OpsNE 100% of the time. If I'm doing it manually, I'm sort of just guessing at some of the best defaults. If I'm doing it automatically, the algorithm is going to pay attention to its progress and the size of the data and the number of parameters and automatically settle those pretty well. So just do that. The other two things that you've got to choose from are whether you want an exact result or an approximate result. At some point, because what t-SNE does is in the high dimensional space, it tries to figure out which cells are similar, which cells are in the parlance of t-SNE or t-distributed stochastic neighbor embedding, which are neighbors, which are very similar across all of the parameters. And I am not going to speak fast enough, for this is going to finish before I'm done. Anyway, so it tries to figure out in high D space what is a neighbor, and to preserve that into the low D plot. And so to figure out what things are similar, at some point, you do have to measure the distance in high dimensional space between all the cells. Think of like Euclidean distance, square root of the squared difference. So exact, we'll just measure all the differences, all the distances. If you want to approximate, as we move through the analysis, we start being able to make some approximations, saying things like, well, if cell 1 and 2 are considered neighbors and 1 and 3 are neighbors, probably 2 and 3 are neighbors then also. And so it is an approximation. It'll make it run faster. It may be less exact, as the name implies, but I've never really seen much of a difference in the appearance of the results, so I tend to pick approximate for the extra speed. The last choice I made was choosing FFT, or fast Fourier transformation, for the gradient descent algorithm. So what all of those math terms mean is that t-SNE is an iterative process, and it tries to make from a completely random start to the t-SNE process, where it'll take the high D, all the cells, and just throw them from high D randomly into the low D plot, and then start to move the ones that are most similar together using probabilities. And each step check whether it's done better or worse. It will do this either with more aggressive step-to-step changes or less aggressive step changes. Your gradient descent algorithm figures out how big a change to make step-to-step. And also things like, maybe I should make big changes at the beginning of running the algorithm and slow it down a little bit as I get closer to a good solution. So the Barnes-Hut was the first gradient descent algorithm that made t-SNE practical for flow data. The original algorithm didn't really have a very good gradient descent whatsoever. It wasn't intended data for data so large and complex as we work with. So it didn't work so well. So Barnes-Hut made it usable. And then fast foray is just, it's a much better approach to gradient descent. It anticipates when to slow down a bit better. So always use fast Fourier, I think, at this point. I will probably in the next version of FlowJo just remove Barnes-Hut as an option, but it's still there for anybody who wants to. So that's the choices I made to produce this graphic. What is this graphic? So it produces two parameters. So t-SNE isn't going to cluster. It's not going to give you any populations all by itself. It'll give you a visualization of all of your phenotypes in one 2D plot. Things that separate out as fairly distinct looking islands, we assume are things of similar phenotype, but we don't know what their actual profile is in terms of intensities of markers until we look at them a little bit. For now, there's just, okay, I see some divisions in my data. You could go and turn on the color axis and say things like, oh, okay, well, I can see I've got a little bit more 45RA expression on the right and a little bit less down here, et cetera. Probably easier than doing this one at a time is in the layout editor, we could explore it. And there's some tools for show me all of the different, all of the different color maps. Let's just grab a new layout, bring in that population. If I right click on it, I can then say, make multigraph overlays, multigraph color mapping, pick a bunch of compensator parameters. See which ones actually have some difference to them. I could probably, if I wanted to spend some time being smart and removing things that I don't think will have any difference because it's either not related to T cells whatsoever or on all T cells, but I'll cut out a few, hit go on the rest. So just as a way of explaining what t-SNE has done here, it's tried to lump things together with similar expression profiles. So we see something lights up is all red down here. So that's NKG2C. So those cells all have that similar expression of that. A lot of dissimilar, a smaller range. I probably could have trimmed out some white space here. Things have a little less CD197 on the right compared to the left, et cetera. So it's trying to just put things together across all the dimensions. So it's a visualization tool. Unto itself, you can eyeball the data and try to understand where there might be differences between samples, but probably the next step to most t-SNE analysis is then to either apply, to draw a circular gate around some cells to see what they are, or apply clustering on them, have data automatically divided up by an algorithm, so then you can try to annotate which populations there's more or less of in your different biological conditions. So t-SNE, primarily visualization tool for looking at high dimensional data on a 2D plot, while still including all of the different phenotypes that might be present in your data. Okay, we are at 2 o'clock here. Are there any more questions in Q&A or the chat? And this is the last chance. We're going to wrap shortly. So if there was anything you want to ask, now is the time to type it.
Maybe just because you're already on this plot, John, there was a question about how these color scales are set.
That is a good question, because I was thinking, as I was staring at it, I was thinking of changing it myself. So the color scales are set here based on the full range of data. So let's put some other parameter on here, perhaps CD7. Okay, so, oh, and that's what, I did futz around with the scales, which is why this is so much higher. So our range goes from blue to red. Anything down at the far left of the plot is going to be blue. Anything up at the far right is going to be red. If everything is sort of in the middle, it all goes green. But you can also adjust those scales using the Customize button. I did futz around with these and made them actually much larger than this cytometer actually handles as a range. Let's change them all at once and watch how that affects our data. Okay. So specifically, we can see the impact of CD7, and that now I have some reddish stuff, CD45R, even more impressively red to, in some cases, blue in various plots. We decided that was probably the best way to do it. Theoretically, we could just take the brightest cell to the dimmest cell, make it go red to blue, but you can imagine for something all negative, you would have a color difference that meant nothing. So it's totally in your control. anything on the right side of the plot turns red, anything on the left side of it, and we're just, oh yeah, 45 array, highly correlated to Disney, left to right, Disney 1. Anyway, so red to blue, and you can control it, trim out the white space to make more dramatic Disney plots. All right, with that, we are a couple minutes over, and there are some really good talks that are going to happen here at MetroFlow in this afternoon that I am going to go attend. So I will thank you for having spent an hour with us. That was terrific of you to join us. That was terrific. Thank you for everyone who typed in a question here. Thank you for everyone that emailed in a question ahead of time. Appreciate knowing that we were going to come in with a few things to talk about and not have an empty chat where we stared at each other. Really nice of you to join us. This will get posted to the web as a recorded webinar. If there's anything you wanted to revisit, my email address is john.flowjo@bd.com. If I stop the share of my screen, that'll bring the website back up. Sorry, not the website, the Zoom background screen back up with my email address right there. Feel free to ask any questions to me. And thank you, Veronica, for moderating. Greatly appreciate it.
No problem. Thanks, everybody.
Hey, have a great day.